Hello, my name is Penpachari Mangmi. Today I will explain the effect of polypropylene microplastic in juvenile zebra fish. The origins of this experiment come from the large amount of plastic waste that enter the environment, such as a river, canal, and oceans. A significant amount of plastic debris falls on the water surface, where it degrades over time and turns into microplastic. Aquatic animals may ingest this microplastic, leading to blockage in their gill and digestive system. Most of the plastic found in the water source is polypropylene because it is commonly used to make everyday items, such as a snack bag and microwave-safe plastic container. You can see its symbol on the packaging right in this picture. In this experiment, we will use zebra fish, also known as Danio radio, for testing. We choose zebra fish because their physiology and genetics are similar to humans, making them useful for further study related to human health. They are also easy to take care of, grow fast, and are not expensive to raise. The objective is the water exposure to polypropylene microplastic effect was evaluated in freshwater organisms of fish. Next, I will talk about the material and method. First, we need to prepare the microplastic. We do this by cutting a polypropylene plastic bag into small pieces, then melting, baking, and filtering them until we get a microplastic powder. Next, we will analyze the functional group and chemical bond of the microplastic powder using FTIR and Raman spectroscopy. We will also check its elemental composition using the EDS method to confirm that the plastic powder is indeed polypropylene. Next, we will prepare the zebra fish for this experiment. We will use fish that weigh about 0.29 gram and have a body length of around 30 millimeters. Then, we will keep them in water that meet the standard measurement set by APSA and feed them once a day. In this experiment, we will mix plastic powder with the fish food and feed it to the fish. The fish will be divided into 4 groups. The control group, which will not get any plastic powder in their food. Group 1, which will get 1 milligram of microplastic per liter. Group 2, which will get 10 milligram per liter. And Group 3, which will get 100 milligram per liter. The fish will be fed this food for 28 days. After that, 10 fish from each tank will be randomly selected for biochemical analysis. After 28 days, 3 fish will be randomly selected from each tank to collect 1 microliter of blood. This will be tested to see what effect the microplastic have on the blood cell. Next, the fish will be dissected, and 2 parts will be used for analysis. The first part is the digestive system, which will be examined for microplastic accumulation. The second part is the liver tissue, which will be used for biochemical analysis, free radical, fat, protein, and free amino acids, and histological. Next, we will talk about the result and discussion. For the first experiment, which is to check for microplastic accumulation in the digestive system, we will digest and filter the digestive tissue and then stain it with nylase dye. The microplastic will appear as red spot. In the control group, no red spot will appear, meaning there is no microplastic accumulation in the digestive system. However, in group 1, 2, and 3, red spot will appear, indicating microplastic accumulation in the digestive system, with group 3 showing the highest accumulation. In the examination of blood cell, it was found that microplastic can cause blood cell to die. There are two types of cell death. The first type is apoptosis, which is when the body signals to remove slightly damaged, deteriorate, or expired cells. When these cells are stained, they will appear yellow. The second type is necrosis, which occur when cells suffer severe damage from lack of oxygen or pollution, causing the cell to die without the body control. This results in the release of inflammatory substance, causing damage to surrounding cells as well. When these cells are stained, they will appear red. Looking at the image, in the control group, the blood cells are green, meaning the cells are normal. In group 1, the blood cells are yellow, indicating apoptosis, which is a level where the body can still manage the cell damage. In group 2 and 3, the blood cells are red, meaning the cells have suffered severe damage and the body is no longer able to control it. The experimental result on free radical reveal an increase in the amount of free radical in the liver. Free radicals are generated when the body exposed to toxin or pollutant. The ingestion of microplastic by fish further stimulates the production of free radicals in the liver. In addition, free radicals also trigger the death of blood cells through both apoptosis and necrosis. The experimental results on fat and protein show that the free radical reacts with fat, forming a toxic substance called marondialdehyde, which damages liver cells and causes deterioration. As shown in the first figure, the amount of this toxin increased in proportion to the amount of microplastic ingested. Moreover, free radicals also damage protein, transforming them into carbonyl protein, which are non-functional. This disruption causes protein-dependent process in the liver to cease. As shown in the second figure, the amount of carbonyl protein increased in line with the amount of microplastic ingested. In the first image, it can be seen that the protein decreased in quantity because it is destroyed by free radicals, resulting in a large amount of free amino acids. This can be observed in the second image, where the amount of free amino acids significantly increased. The examination of liver tissue layer revealed that in the group exposed to microplastic, there were increased space in the liver cell due to cell death. Additionally, there was increased presence of white blood cells in the liver as they attempt to repair the damaged area. In conclusion, the experimental results show that polypropylene microplastic can accumulate in the digestive system of zebrafish and adversely affect liver function, leading to liver cell death. This results in a weak body and reduced immune response in zebrafish.